important the bioinformatics, life sciences, and the different areas are utilizing sources from these databases. These are the NCBI and the Ensemble genome browser, and C bioportal is utilized for the cancer genomics, and Oncomine is also utilized for the mining of data about different types of cancer. ENCODE is also helping for the analysis of genome in the humans. And the bioinformatics tools that will be utilized today by us are the NCBI Primer Blast and the Primer Quest and the Oligo Analyzer. And we will also see one new software, SnapGene, that I utilized throughout my career, and it is very beneficial. Though it is paid one, but its free version is also available that gives some limited facilities. We all of us know about what is PCR. It is utilized to make a huge number of copies from very small quantities of DNA. Even a few nanogram of the DNA can be utilized to make millions of copies in a matter of hours. Like 20 cycles can turn one copy of DNA into 1 million copies. There are 3 steps of the principles of PCR that are shown somewhere here, and 2 extra initial starting step and 1 is extra and the end step are added along with these 3 cycling steps of PCR. These are melting step, and the next one is annealing of the primers, and we will be talking in detail about this one in today's presentation. And the third step that is also the principle of PCR is the extension step that is performed at 72 degrees centigrade. If this step is longer, like 18 minutes, for longer extensions of DNA fragments, sometimes we decrease this temperature up to 70 degrees centigrade and even up to 68 degrees centigrade in some cases, but usually we do perform usual routine PCR at 72 degrees centigrade. And some of the fragments that miss the opportunity of full extension are given an extra extension time of 5 minutes, and at the end all the product, the final reaction, PCR reaction, is stored at 12 degrees centigrade in the machine. Okay, so some of the resemblance I always give, but I will take very short time today about the introduction. That some of the guy has turned on the speaker. Please turn off, the sound is bouncing back. Okay, so deionized water, the PCR buffer, magnesium chloride, DNTPs, DNA polymerase, DNTPs, primers, DNA template, and dye. All these are usually the component of a PCR. In these things, first, second, and third one are similar to the cement that a mason is putting to build a wall, and the DNA polymerase is similar to the mason that is building a wall, and the bricks are similar to the DNTPs that are utilized, and the brick and the wall is then built. And you know, the primer provides the basis, the starting and the end point for the extension of a PCR. This is somewhat similar to the bases that are built before building a wall. And the template DNA is the whole land that cannot be amplified until the primers are provided. And dyes, some of the PCR are requiring are added with the dye as an extra thing. But the salmon, these are the essential element for the PCR. All of you know the pH and the requirement of PCR buffer, DNA template, magnesium chloride.
Excuse me, sir. Sir, there is a distortion in the voice. Please check the
Okay.
mic. Please check the mic. There's a distortion,
Okay.
and the voice quality is not very good. So participant asks to be a good voice quality from you. So please check your hand-free or mic for better communication. Thank you.
Okay. I speak again. Please check. Is it better now? Is it better? Can you hear
Yeah,
me?
it's much better now. Yes, it's much better, sir.
Okay.
You can continue now.
So I hold my mic with my hand, so it will be better, I think. Okay. So
Yes,
the
it is.
DNTPs
It is.
and the DNA polymerase. Okay. Thank you. Okay. The DNA polymerase. So today we will the target point today. So what the primers are? Primers are actually the nucleic acid strands that provide the starting and the endpoint. They are usually added to the PCR reaction from 0.2 to 1 micromolar concentration. Usually they are 18 to 25 nucleotide long. And they must have some specific attributes, like they should have 40 to 60% of the GC contents. If the GC contents are too high, the primer will bind non-specifically to other places other than our required. And if the GC contents are very low, they will have the poor binding and poor amplification of our DNA fragments. And the primer must not be self-complementary. If they are self-complementary, they will bind to each other and they will not bind to the target sequence, and there will be no amplification. And the primer must not make the hairpin. If they are making the hairpin, they will not bind to the target sequence. Not more than 280 bases should be at the 3' end. If this is 5' end of the primer, and if this is the 3' end, there should not be more than 280 bases. Usually it is recommended at the 3' end. If there are more than 2 or 3 or 4, 5, 6, 80 bases that have the double hydrogen bond, the primer will have the poor binding. So 80 bases have the double hydrogen bond. And there should not be more than 3 CG bases at the 3' end. If there are more than 3 CG bases, that
I think there's some technical problem. So please be patient. He will be back soon. Thank you. Participant, please be patient. He will be back soon.
Hello.
Sir, you are visible and audible now. Sir, okay, you can start.
Can you see my slide?
I can't see your slide, but now it's visible. You can start the session now.
Okay. So I'm really sorry. I missed my connection. I don't know why it happened. So everything. So where I was at somewhere at the hairpin. Is it right?
Yes, you are presenter now.
Okay. Okay. So I was talking about the forward and the reverse primer. The forward primer is simply the 20. It's designed by taking simply the 20. It's designed simply by taking the 20 mers from the top strand, and the reverse primer is designed by taking the 20 mers from the top strand and by taking its reverse complement. By taking the reverse complement, we design the reverse primer. The forward primer binds to the bottom strand and the reverse primer binds to the top strand. You can see here the three steps that are repeated 20 or 30 times in a PCR reaction. At the 95 degrees centigrade, the double strand melts. And at the 55 to 60 degrees centigrade, both the forward primer and the reverse primer binds to the sites of complementary. And at the 72 degrees centigrade, the enzymes comes in action. They hits at the site of binding of primer, and then they start adding the DNTPs from the 5' end to the 3' end. In the same way, the bottom strand is extended. So you can see here the extended fragment. Okay, now we move to the real portion. Okay, so we are going to use some of the software for today's presentation. So today we will be learning how to retrieve the data from the NCBI and Ensemble. I will let you know benefits of each of these two databases. We will learn today designing of primers by the NCBI Primer Blast. We will also see the designing of primer for the qPCR by the Primer Quest. We will also see today the use of SnapGene for the designing of primers. We will also see today oligo analyzer, and we will analyze the primers for their quality. So let's start one by one. So the primer designing for the endpoint PCR. Endpoint PCR is a simple PCR that we perform every day in a simple PCR machine. It is called endpoint because we see the result at the end of the PCR, like after 2 or 3 hours after the completion of 20 cycles or 30 cycles. So first of all, we need to have the sequence of the DNA for which we want to design the PCR primers. Every one of us know about the NCBI. So for the designing of PCR, first of all, we are going to retrieve the sequence, the DNA sequence, the gene sequence of EI24. We go to the NCBI software first of all, and then we select the gene here in the drop-down menu, and then we write the EI24 in the blank blank enter. After we hit the enter, we come across a new window that is giving the different EI24 sequences. Among these sequences, we have to select the EI24 that correspond to the human EI24 gene. This is written Homo sapien here. So we will click this one, first one, EI24. After we click this one, we go and we click the Gen Bank. After clicking this Gen Bank, we come across a new window that is containing the information about this gene. For example, this gene has the NC number, this one, locus number NC 00011. And this one is KB, 15 kilo base pairs, 15,474 base pairs. And this sequence was submitted on the 6th June 2016. And if we go a bit down, we come across it's which kind of transcript variant it is. We have to select many times transcript variant 1 because in humans, in animal, the transcript variant 1 is predominantly expressed. Sometimes transcript variant 2 is predominant in its function, and we have to select the transcript variant 2. So we have to see which kind of information when we are retrieving the sequence. First of all, we have to check its name, and then we have to check its transcript variant. And also we have to know the M number of its coding sequence. This is also very important. Transcript ID, this is called. And here we click on the CDS. This is called the coding sequence. If we want to find the coding sequence of the EI24 gene, we click EI24 at the start. We have to click the CDS. After we click the CDS, this new window, this happens with this sequence. This is actually the transcript of the EI24 gene. This is containing the UTR upstream untranslated regions and downstream untranslated region. And we are interested only in, for example, the coding sequence. So the identification of coding sequence is this one. After clicking the CDS on the NCBI, this coding sequence become highlighted, and its identification is this one. It starts from the ATG and it stops at the TGA or TAG. TGA is a stop codon and ATG is start codon. We copy this sequence for the designing of primers. We copy from ATG to TGA. This one is the copied sequence from ATG to TGA. We paste it in a Word file for our record. And we open now the NCBI Primer Blast. We go to the Google and we put simply the NCBI Primer Blast. After we open this window, this is the web address of this website. So can you hear me? So should I continue? Can you hear me?
Yes, Dr. Muhamma, you are audible. Okay. Please continue.
Okay, thank you. I will check several times, I'm afraid. Okay, so we will copy this sequence, this whole sequence, and we will paste in this blank window, Okay, so this website is opened, and I will try to share the video recording of this lecture on the website, and you can follow the simple steps for the designing of your own primer. I will try to make it sure for the recording and for the sharing on the YouTube channel or somewhere. Okay, so the sequence will be pasted somewhere here in this blank window, and rest of the parameter we have to give. Let's see on the next window. So this sequence has been pasted, and we have to give the number of base pair from which the forward primer will be designed and for which the reverse primer will be designed. For example, if I say forward primer must be designed from 1 to 700 base pairs, and reverse primer is designed from the 300 to 1023 base pair, because this range is wrong, so software will not accept this range. So this forward and reverse primer region should not be overlapping. So I correct my range. I gave the range 1 to 500 base pair for the forward primer and 501 to 1023 base pair for the designing of reverse primer. And rest of the parameter I should also fix. Those parameters are all already given by the NCBI, but I should make sure about the rest of the parameter. Forward and reverse primer regions, and what should be the minimum and maximum length of the PCR product. I say that PCR product must be between the 100 and maximum up to 300 base pair, the PCR amplified product. And I say that the PCR for primers, 10 primers, please return me 10 primers, please return me 10 primer pairs. And I already talked to you, I like the optimum annealing or melting temperature to be 60 degrees centigrade. I say optimum melting temperature should be 60, and it should be minimum 57 and should be maximum 63, and the maximum TM difference should be 3. And after giving all these parameters, I say click yes, and after I click yes, this kind of processing starts. And after this kind of processing starts, we click somewhere on this NM number. I already talked that we need the coding sequence several times during our experiment, so we record this NM number for the gene of our interest, that is EI24, and that corresponds to Homo sapien humans, and it's transcript variant 1 in which we are interested in. So we will click this one NM number, and after clicking this one, this NCBI primer blast return as 10 primer pairs, and the first primer pair is somewhere here. This is forward primer F1 F2, this is F2 R3, F1 R1, F2 R2, F3 R3, F4 R4. And usually the first primer pair is best one. And the sequence of all these designed primer pairs that has been shown graphically here, and these graphically shown primers sequence of these primers are given in the bottom. Usually the first primer is the best one. For example, this primer is, forward primer is this one, and reverse primer is this one. Forward primer binds to the plus strand and the reverse primer binds to the minus strand, and they both do have the 20 mer of the length, 20 bases long. These primers are, they start at 386 forward primer, and the forward primer ends at 405. In the same way, the start of reverse primer is from here to here, and the TM of the forward is 16 degrees centigrade, for the reverse it is same, and GC contents are falling well within the range that is between the 40 to 60% the GC content should be, and it is exactly fulfilling our requirement. And it is going to amplify, that is going to give the 2 to 3 base pair of the product length after the completion of PCR. In this way, we simply design the endpoint PCR by the use of NCBI primer blast, and I used during my career this software several times, and I got success. Even I obtained the 6 KB 600 base pair of the product length by using this software. This software is very efficient, and I got success using this software. Next, we are going to design the primers for the quantitative PCR. In the previous endpoint PCR, we obtained the DNA sequence from the NCBI, but this time we are going to use another database that is called the Ensemble. This is very useful database that is containing the DNA sequence, gene sequence, mRNA, transcript, and protein sequence for human, for mouse, and for several other species. Simply you will write the Ensemble genome browser and you will come across this website. And we are going to design the qPCR primers for this EI24 gene, EI24 mRNA, that is for the human. So we'll write the species name in the Ensemble website, and we will write the name of the gene, and then we will click the go. After clicking the go, we will come across a new window just like in the case of the NCBI. So they search 6 gene against the EI24 name, and 19 is searched and against this one, and gene tree 1, and several other features that search for the EI24 gene. And we are interested in this EI24 gene, that is the human one. So we will click here on the first one. For example, we can click on the first one. After clicking on the first one, we will say the transcript table, because several genes have different transcript variant, and in the previous case we selected the transcript variant 1 because its transcript variant 1 is predominantly expressed and is predominantly performing the function. If we by mistake select the transcript variant 2 for the EI24, that is not going to give the phenotype, the results, and even if we work for 10 years, it is we are going nowhere. So we have to design the experiment very carefully. We have to select transcript variant carefully, cloning, and when we are designing the primers. Okay, after clicking on the transcript variant table, so this transcript table is appearing. The first one, this one, EI24201, but the NCBI was showing transcript variant 1. Here it is written 201. And this is the NM number that will be beneficial in this case. We will note down this NM number. NM number is NM_004879. This NM number will be utilized to retrieve the sequence and to design the primers for the qPCR. And I let you know some of the attributes that are bit different from the endpoint PCR when we are designing, especially in the qPCR. Okay, we will click at EI24/201. So this kind of transcript sequence is opened. This is upstream UTR and this is ATG. This is the start codon, and for every start codon, it is going to give the amino acid. Amino acid against each codon is shown here. This kind of facility is not in the NCBI, but Ensemble is giving this kind of annotation, this kind of information. Okay, we can configure this page and we can select the only show exons and don't show in the lowercase and show codons, just show codons and show UTRs and show this kind of. We will select only the first one and we will unselect all of the rest of the things. When we will select first one and we will click OK, so we will come across this kind of sequence. And this is very beautiful actually. This facility even is not available in the NCBI when we obtain the sequence of the mRNA of the EI24. This is the coding sequence. This is ATG, this is TGA, this is stop codon. And you can see some of the bases are shown in blue and some of the bases are shown in black. These blue and black are actually the alternative exons. These blue and blacks are actually the alternative exons. And the PCR primers, we already know that designed in a way that forward primer should be on a different exon and the reverse primer should be on the different exons. If they are falling on the same exon, some of the DNA contamination during the RNA extraction may be giving the false positive results. If the reverse and forward primers are designed on different exons, for example, E1, this is E1, and this is the intron, and this is the exon 2, E2. If the primers are designed qPCR on different exons, then the forward and reverse primer, even upon the presence of the DNA contamination, will not give the false positive results because this span is so long, this will not be amplified during a 30-second extension. For example, in the qPCR, we give 30 to 40 second or maximum 1 minute extension time. So that's why that they are designed on different exons, and we can see clearly that different colors are showing the different alternative exons. Okay, but we are not going to design these primers manually because we are talking today about the tools for the designing of primers. We already noted the NM number from the Ensemble. So now we are going to the PrimerQuest freeware online software. We search from the PrimerQuest software, and this is the PrimerQuest. This is the website itDNA, and you can see here. You can click on the tools and then you can click on the PrimerQuest tool. You can click on the tools and you can click on the PrimerQuest tool. After clicking this tool, a new window will be visible. This is giving the different options. This is PrimerQuest tool. Either we can enter the sequence manually or either we can download the sequence from the GenBank by giving the NM number, by giving the accession ID. We will click on this facility. We will click on the download sequence. After clicking on the download sequence, a new window will be open and we will simply paste the NM number. After pasting the NM number, we will click the get sequence. After we click the get sequence, this sequence will automatically be obtained by this website from the NCBI. And after clicking this one, we will click on the qPCR primers, two primer with intercalating dye. We have two options: either we can design primers with two primers with probe, or we can design primers only two primers that have the intercalating dye option. Okay, usually we design this kind of qPCR primers, and this kind of qPCR primers are utilized in diagnostics. Diagnostics cannot be authenticated until the probe is included along with the in the qPCR. Okay, we are going to simply design this option. We click this one, and we will show custom design parameters. Sometimes we click and we don't need, actually they are already well set. We will click here, and after clicking, we will come across different qPCR primers. You can see some are here, some are shown here. First forward primer is going to bind 319 and 342. It is stopping. It is 23 base pair long, and TM is 62 degrees centigrade. If its TM is 62 degrees centigrade, it means it should bind at 60 degrees centigrade. At the start, I said I like those qPCR primers that bind at 60. That bind at 60 means it has annealing of 60 degrees centigrade. It has annealing of 60 degrees centigrade. TM will be usually 2 or 3 degrees centigrade higher than the annealing temperature, and GC contents are well within range, 47.8% and the 50% for the reverse primer. To see the sequence of primers, we will click on the view assay detail. After we click the view assay detail, we come across the sequence of primers. Not only we come across the sequence of the same primers, but we will also see where these primers are actually binding on the mRNA. If we copy this sequence and we search the sequence on this table, we come to know that the forward primer is binding on the exon 1, 2 on the second exon, and the reverse primer is binding on the third exon. So this kind of selection that the primers, forward and reverse primer should be on the different exon has been done by this software, and you can see here they are designed on a different exons. Because we gave the NM number, and by giving the NM number to this PrimerQuest software, so they already obtained all the information of the, not only about the sequence, but about also about the exons, and they designed on different exon the qPCR primer for us. Okay, so one more software I share with you today, that is SnapGene. That is very useful. Many times it is said that we need to design sometime the softwares by the by manual method. So this is the manual along with some extended facilities. This is the SnapGene that I have purchased during my PhD, and I brought here back in Pakistan. This is very useful for the designing of primers and several other options. This is SnapGene, and its free viewer mode is also available. This is active for 1 month, and we can design good software by using this good primers by using this software, and then it is only comes in the viewer mode. So limited facilities are available only for 1 month as free. So by opening this software, we click on the new DNA file, and we copy this ATG from ATG to TGA, all the base pairs of the EA24 gene, and we click new DNA file. After clicking on the new DNA file, we paste the sequence, and this software asks, please remove the non-DNA characters. We can paste any non-DNA sequence. Okay, we will click OK, and this kind of 1023 base pairs are pasted in this empty window. We give this sequence a number like human EI24 coding sequence, we can say. Okay, and this is 1023 base pair long, and I added the name, and we will click OK. After clicking OK, we come across this kind of stage. This is 1023 bases. This is 1023 base pairs, and you can see different restriction enzyme sites have been shown. These are very useful in the designing of cloning, in the designing of constructs. in cloning strategies. Okay, so we will click somewhere here on the sequence. This is the map. This stretch is showing the map and the sequence. After we click on the sequence, we come across this kind of sequence. If we select this much sequence of the DNA, so the software automatically calculates this is 16 base pair long and this is 51 to 52 degrees centigrade and its melting temperature is. And you can see here the 50% GC contents are there within this selection. Okay, so sorry. So we will select this sequence and we will say add primers. After selecting the sequence, we will say add primers. After we say the add primers, the new window will be opened and it will ask you have to say I have to design the forward primer that is on the top strand. We copy the sequence simply from the top strand, that is called the forward primer. After clicking the top strand, the new window opens and this top strand is selected. Okay, this top strand is selected and we can see if we have to do the cloning, sometimes we have to add the restriction sites. For example, we want to add the restriction site at the 5' end. We want to add the restriction site at the 5' end, and we have to select the BAM-H1, for example, and we will simply click the insert. After clicking the insert and after by putting the cursor or on the 5' end, the BAM-H1 site is introduced. And you already know not only the restriction sites is added, but some extra bases, 4 to 6 bases are added because the enzymes cannot cut when they are sitting on the edge. For example, this is the restriction site for enzyme BAM-H1. I try to write BAM-H1. For example, this is the BAM-H1 restriction site. So it will not cut until it has some edge here, some extra bases. It cannot sit on the edge and it cannot cut somewhere here. So we have to add some extra bases that are shown here in the small letters, and these are the regular letters. Regular letters are showing the restriction site, and these are the letters that are the bases for the primer. And we will say simply add primer to the template and the forward primer is added. And you can see this forward primer is added here. For the designing of reverse primer, we go to the bottom strand because for the designing of the primer, if we want to clone this whole coding sequence in a new construct, we want its expression in the mammalian or in the bacterial expression system, we have to clone not only the start codon up from the start codon up to the stop codon. We simply select these 20 mers, for example. These are 23 base pairs. So software automatically let us know that the TM temperature is 60 to 60 degrees centigrade and there are 52% GC content. This is also falling once again within range. So after selecting this one, we select once again the add primers. Within the SnapGene software, we select the add primers here. After clicking this one, so software is again asking where you want to design, on the top strand or bottom strand, because this is reverse primer. We have to click the bottom strand. After clicking the bottom strand, we put the cursor on the 5' end of the primer and we select the EcoR1 from this drop down menu and we select the insert and we add the 4 extra bases on the 5' end of the restriction site. Now small letters are extra bases that are shown in red, and the capital and red color bases are the restriction site, and the black and capitals are the reverse primer, sequence of the reverse primer. We can even add some of the codon here by selecting from here. Because this is the reverse primer, we have to select the reverse complement here. We have to select the reverse complement. For the basis of the reverse primer, it does the reverse complement automatically, but for the reverse complement of the restriction site, and you can say somewhat about the extra bases. We have to select the reverse complement, and we will say add primer to the template, and in this way the reverse primer is added. Then we click on the primers. After we click on the primer sequence is visible somewhere here. So I named the forward primer BAMH1 because it has the BAMH1 restriction site, EI24 gene, and this is forward primer. And for the reverse, EcoR1, EI24, and reverse primer. This way we selected the primers for the full length EI24 coding sequence by the use of SnapGene software. Next, we are moving for the testing of primers. Primer testing is very important. Even if we download the primer sequence from some of the papers, we must test these primers whether these are good or not. So there are two kind of testing of the primers. One testing is that whether the primers are binding on the gene of our interest or not, and second testing of the primer is that whether they do have the enough capability to bind the site of their complementarity or not. So two kind of testing we will do today before we perform the primers. This will also be this kind of testing will be performed in silico. First testing is for the complementarity, whether they are complement to the site of our interest or not. We should test. So for this sake, for example, we took two primer pairs. These are three primers here shown here. These are also published in my experimental manual. And these are the real sequence of primers, and these are also real sequence of primers that are utilized. Okay, so the NCBI primer blast, we will write NCBI primer blast, and we will click this first one. And after clicking this first one, in the designing of primers, what did we do? We did enter the sequence of DNA for which we want to design primer, but in this case, bit different strategy is obtained. We'll put the sequence of forward primer here, and we'll put the sequence of reverse primer here. We don't put anything here. We don't put anything here. We put the forward sequence on the place of forward primer, and we put the sequence on the place of reverse primer. And this is again the website. This is the primer blast that is also utilized for the designing of primers. This will be utilized today, and you can see for the testing of primer for their site of complementarity. Okay, after we put the sequence, you can see here, and what do? We say simply get primers. We will simply click get primers, and the now the primers have been obtained. We put the same sequence of forward primer, and we put the same sequence of reverse primer, and we come across that these two primers are binding here and here. And you can see here that EI24 primer was designed of EI20 transcript variant 1. Okay, and this one is also a similar sequence. Okay, this is the TERT, and this TERT mRNA. This is TERT because first letter is capital, rest of the letters are small. It shows this correspond to the mouse. This correspond to the mouse. If it were human, all the letter must be capital. This is the notation for the gene, that all the letters are capital for human, and for the mouse, first letter is capital and rest of the letters are small. And this is, you can see, musculus. And this one is going to give the product length of 69 base pairs. And in this way, we checked that whether our primers are binding to the site of their complementarity or not. We checked the TERT primers. You can see sequence here. This one sequence, GCGTGG, GCGTGG. This one was the first forward primer, and this was the reverse primer for the TERT. And we checked they are binding to their exact site. They do have the complementarity. Okay, they are binding to the gene of our interest. So the next testing comes across. So next testing is attribute of the primer. How much strongly it can bind, how much perfectly it can bind to the site of its complementarity, whether it is loose binding primer or the strong binding primer. Loose binding primer will give the low amplification. Okay, so here in this case, we will use again the oligo analyzer. This software needs a short sign up. It's no problem. We can do this one, and this is the address website. We go to this page, oligo analyzer. Okay, and then we click here, the start using oligo analyzer. Start using. Previously we went into the tools for the designing of qPCR primer, but for the testing of primers we will go into the oligo analyzer. After we go into the oligo analyzer, we will sign in here. After we sign in here, so it does have the two kind of capabilities. It can test a single primer, or it can design the both primer together. First of all, we will learn how a single primer is analyzed. We will paste the sequence here and we will simply click analyze. So what does it give? It gives the different concentration required for the amplification. It's okay, we already know. It gives a sequence of the primer once again, and it gives the complement of the primers. It gives how long it is, 26 mR it is long, and it is containing 53.8% of the GC content. Its melting temperature is this one, and molecular weight is this one, and the extinction coefficient is this one, and different details are given by clicking on the analyze. This is single primer, we paste it here. Next parameter that we will test for this single primer is hairpin. Whether this primer is making hairpin or not, we will click on the hairpin. After clicking on the hairpin, this is amplified version of the previous slide. So different hairpin informations are here. It is giving the TM for the hairpin. TM for the hairpin should be lower than the TM annealing temperature of the primer. If the TM is higher than the annealing temperature, it will predominantly make the hairpin. It will not give the amplification. Moreover, it is also giving the delta G value. If the delta G value is very low, it means it can spontaneously make the hairpin. We already know from the thermodynamics of the reactions. If the delta G is very low, so the delta G should be higher than -6 kilocalorie per mole, and this primer is having minus 3.54 kilocalorie per mole. So these primers will not predominantly make the hairpin because they need bit higher bit higher energy. So if the energy is very low, then they will make spontaneously automatically. So the next parameter is the self dimer, whether they are making self dimer or not. So we check here, they are only making self dimer at 3 places, very low. So it is good, this primer is good, this primer is not making the self dimer. Now we are going to test the two primers at the same time. So we will paste the forward primer here, we will paste the reverse primer here, and we click on the heterodimer. We click on the heterodimer. Okay, we click on the heterodimer. After we click on the heterodimer, it is calculating where they are binding to each other. Only 4 places. It is very low. It's very low, and this is also giving the energy for the formation of heterodimer. If the heterodimer energy is very low, it means they will easily make the heterodimer. And you can see this is 6.24 kilocalorie per mole, and if the energy is greater than -7, then it will not make the heterodimer. The energy is greater than -7, and it is well above the -7. It is -6.24. So these two primer pairs will not make the heterodimer. This way we tested the primers. So these are all things, and at the end I introduce you with my group. These are all the students of my research group, and some of these students are working on the diabetes, some are working on the liver injury, some are working on the production of monoclonal antibodies by the recombinant DNA technology, and some are working on the hybridoma technology for the production of monoclonal antibodies. And these are some of the projects that we are running in our labs, and for the completion of these projects we developed the cell culture lab that is fully working. And this is my student Safdar. He is well expert in the cell culture technology. We have also developed the mouse room at the Laboratory Animal Research Center for the in vivo studies, and this is fully functional here. And we also developed the molecular biology lab, and this is she's the Rida. She already graduated, and she also about to publish one article. It is almost accepted in the molecular biology reports. She did work very hard. The Rida and the Safdar, they do have they are the co-author of the publication. And this is our institute, and this is the Quwwat Faisalabad Institute of Research, Science and Technology. This is very beautiful. This is 33-acre area, and We do have the girls hostel and boys hostel inside. We have the mess rooms, library, internet facility. We do have the sports grounds, more than one, and we do have the basketball and the badminton courts, and we do have the biotechnology lab, biochemistry lab, the molecular biology, cell biology, in vivo studies, the tissue, plant tissue culture, the microbiology lab is also very important here in Hoover Faisalabad Institute of Science and Technology. And thanks for listening. So if you have any question, you can ask uh, about the primers, about the designing of primers, about the attributes of the primers. Thank you very much.
Thank you so much, sir, for such an informative session on primer designing. Hope you all get the information and the concept you want to get. Uh, Now we'll go ahead to take some time for questions now. Just a reminder, please be sure to raise your hand. I will take your question by unmuting you. So another thing you have to be make sure that your question should be small and precise so that the presenter can understand and answer you. Okay. So it looks like we have a lot of questions by raising the hands. So everyone be patient for their turn. I will take some questions. I will not take all the questions, but take some questions. So don't worry about it. You can share your question in the discussion forum in our Facebook page, where Sir Dr. Muhammad Ali can answer you, I think. Sir, can you cooperate in
Yes,
our
yes, I can hear.
Facebook page? Sir,
Inshallah,
can you?
inshallah. Definitely, I will.
Okay, okay,
I will
okay.
do. I
Thank
will
you so
do.
much. Okay, the first question we will take from Kashif Hussain. I allow your mic now. Please unmute yourself if you want to ask the question. Kashif Hussain, can you hear me?
Ma'am, I asked a question that how can we isolate a primer from bacteria or agrobacteria, and what's the major methods and what are the material we use for that?
Okay.
So, okay, so I cannot fully hear. So this is question is out of the context of this presentation. But we can isolate different bacterial strains even from the soil, even from some old samples. We can separate. We can collect the soil sample or some old sample, and we can grind, and then we can spread on the plate, and then we try to make the single colony, and that single colony is then identified. This is specially a special question for the microbiology actually. Okay, so we can identify on
Okay.
the basis of their morphology, and then also on the basis of their genome. We sequence their ribosomal sequence. We get their ribosomal sequence, and we send for sequencing. On the basis of their sequence, we identify different kind of bacteria on the morphology and on their DNA sequence. Yes.
Oh,
Next,
okay.
please.
Okay. Now, next question is from Afra Sayab. Afra Sayab, you can unmute yourself and
Yes.
ask the question. Afra Sayab, can you hear me? Afra Sayab. Okay, move to the next. Noreen Naz.
Yes,
Okay,
please.
Noreen Naz, can you can ask the question. Noreen Naz, you can unmute yourself and ask the question if you have. Okay, I think she's not present. Okay, Parveen Kumar. Parveen Kumar, you can ask the question.
Okay. Sir, I have two doubts, sir. First one is why should we select a transcript variant of one?
I can't hear you. Please Can you ask us again? Pardon, please.
Okay, ma'am. Can you able to hear my voice, ma'am?
Yes, but there's some disturbance in the voice as well.
Why should we select a transcript variant of one?
I can't get your question.
So I type in the
Have
chat, ma'am.
Have you got the question, sir? Dr. Muhammad Ali? Okay, there is some problem.
Is it audible, ma'am?
So we have some technical issue again. Okay, can we have Dr. Zaman Tariq? Dr. Zaman Tariq, he is an advisory board member of World Forum of Young Scientists. Can you hear me, Dr. Mahmoud?
Hello? Yes,
Yes.
I can hear you. Okay.
Okay, we have some questions. I think
Yes.
Parveen Kumar, we can't got your question. Sorry for that. The voice is not clear. And now we have Wajiha Afridi. Wajiha Afridi, you can unmute yourself and ask the question.
Yes, Wajiha Afridi.
Wajiha Afridi, can you hear me? Oh, I think she's not present. I don't know what happens, why they are raising their hand if they don't want to ask the question. Please raise your hand down if you
Right.
don't want to ask the question, please.
Maybe sometime they write questions and we ask from their behalf. Sometime it happens, but today they are, I think, some problem in their internet, maybe.
Okay, Amna Chaudhary, you can unmute yourself and ask the question if you have. Amna Chaudhary. Amna Chaudhary. I think she is also not present. But I think if they are writing some
Yes.
question in the chat box.
Yeah, I'm seeing if there is some question.
Okay, I also see.
Okay.
Okay, meanwhile, there is a feedback form you all have to fill there,
Yes.
where you can get e-certificate. Okay.
So please
Ma'am,
share
please
the
tell
link.
me
So
again.
Okay. Okay. Who's this?
Kashif Hussain.
I'm Kashif Hussain.
Okay. Okay. Kashif Hussain. You can ask the question, Kashif Hussain.
Ma'am, you already said that the procedure to get e-certificate and outside. How can I get this one?
You can get your
So the matter.
you can get your e-certificate by registering, but you can also have to fill the feedback form, which I have shared the link in the chat box. You can see it. Okay. The major advantage of the feedback form is that we want to improve our webinars. How can we improve our webinars? What new thing we can add in it? So this is the way to get your feedback. So it is good to have your feedback. Thank you. Okay, now, sir, can you see any question in the chat box?
Okay, I'm trying to. I just shared your given link once again. So all the students should fill this feedback form to get the e-certificate. If you miss, maybe you will not get. I'm not sure. So okay,
Okay.
I'm trying.
Okay. Why does NCBI Primer
Okay.
Blast design primer only for limited region in the transcript? This is from Myra Sumro. Sir,
So why NCP primer blast designs primers?
only for a limited region
Okay.
of the in the transcript.
No, it does not design for the limited region. We have to give the limits actually, because we want that our primer length should be from this to this, and also our primer should be from this to this region. We have to give limits to the software. Otherwise, it does not. He does not know where to design the primer. So it is for our benefit actually. And also, why did I show only 3 softwares? Because these are the 3 softwares that give me success always. So I'm actually sharing through this presentation my success stories with you. I'm using these softwares, and I use these softwares with great success, and I want you to be successful. That's why I'm sharing my ideas. And also I have written some of the books about the molecular biology, and I share maybe its picture on here with you. uh, so some of the experimental manuals I have written, and I share their title with you here. So you can go through my manuals, and you can ask question on the Facebook page, as respected Dr. Saba asked. Where these are the actual books. You see my screen? Can I?
You can share your screen first.
Okay. Can you see my screen? Right. So this second one. So this second one is the experimental manual that is containing the designing of primers by using the NCBI and the primer paste, and different strategies are given. And this is more like the molecular biology and cell biology books, and the 80% of the protocols in this manuals are from my PhD study. During my PhD study, whatever did I do and whatever obtained I success in, I shared in this manual, this green one. And this first manual that I wrote already, that is already taught in different universities, UF, Minhaj, Fajafrit, etc. This is more like biochemistry. This is containing experiments for the 5 to 6 courses, and this experimental manual is containing the practicals about 3 to 4 courses. And this one is going to be published, and I'm writing this manual. And this is more like about the clinical and the metabolism. So today I presented most of the things from this green manual. This is already published, available in market in Faisalabad. So anyone, any question, please ask.
Okay, I'm going to. Can we carry out some part of the protocol in dry lab in silico? This is
Yes.
from T. Satyanarayan.
Like in silico is the first step before we move to the wet lab. So only dry lab is not going to give us much benefit until we perform it in the wet lab. And one more thing, dry lab is not confirmatory always. It's not 100%. It's like 80%, 90%, 95%, 98% confirmatory. The in silico studies, whatever we design today, most of the time we design 2 or 3 primer pairs. Even if we test the quality, still we design 2 or 3 primers, and some of primer may not be good even after using in silico studies. So that's why the wet studies, the wet lab is mandatory. Another question, please?
Okay, this is from Rao Zubair Khalik. He is asking that
Yes.
shouldn't we design the primer manually in order to meet our requirements, that is melting temperature, primer size, and GC content?
We can design manually, but our brain cannot analyze all the parameters. Which parameters? Like our brain cannot. First of all, it can only see 20 merge. It should be long. Primer should be 20 merge long. Our brain can think this way, but our brain cannot think. Okay, 40 to 60% GC content. Okay, it should not make hairpin. Okay, it should not complementary. Okay, it should not bind to 10th chromosome or 15th chromosome. Our brain cannot, but software does everything for us. Okay, forward reverse primer should not have melting temperature differing from each other more than 3 degrees centigrade. Our brain cannot very easily analyze all these combinations, all these attributes. So that's why we get help from the softwares. I usually teach about manual primers and different cloning strategies, but today I did not do because today I was majorly using the softwares, and software can help us, and we cannot think all these parameters. That's why we always get help from the software. Yes, another question, please.
Okay, there is an announcement for the participants that you please don't worry about the e-certificate. The
Yes.
e-certificate will be issued later on your emails. So don't worry, just fill the feedback form and get your e-certificate in your emails. Okay? So don't worry about
So
the
what's the
e-certificates.
limitation of filling the performer? Please share with the students. Like within 1 hour they should send, madam.
No,
Please
no,
let us know.
it's you just after the meeting the link I think is not valid for access, I think. So I think you should fill the form within some minutes of
Now.
presentation, I think. Okay, please arrange other webinar on machine learning or artificial intelligence based drug design. Okay, thank you for your feedback, but you have to write this feedback on the feedback form, Mr. Muhammad Danial. Okay, why should we select transcript variant 1? Can you hear me, Mr. Muhammad Ali? There is another problem, I think. Mr. Muhammad Ali, can you hear me? Okay. Meanwhile, I have to. I would like to invite Mr. Dr. Tariq Zaman. Sir, can you hear me, sir? Dr. Tariq
Yes.
Zaman.
Yes.
Okay, sir, it was just 1 minute. I have a call from the speaker. Okay, there is some technical problem with Dr. Muhammad Ali, so he called me, so I just turned his call. Okay, now move to the Dr. Tariq Zaman. He is he's our advisory board member of World Forum of Young Scientists. Currently, he's in USA. Am I right, Dr. Tariq Zaman? Welcome to the
Yes.
seminar.
Yes.
Sorry,
Yes.
sorry, I got you after a long searching in the participant, because we have got a very good response in this session, and we have got more than 300 participants and got more than 400 registration for the seminar. So it is bit difficult for me to I didn't recognize you for the for some talk. Okay, Dr. Tariq Zaman, he is an internationally trained molecular and cellular neuroscientist. He investigated physiological role of sodium, calcium, and calcium-dependent channels in absence epilepsy, early infantile epilepsy, sensory filtering related to autism, and neural control of obesity. Right now, he is a research professor in College of Human Medicine, Michigan State University, USA. So, welcome to the session, and please tell us something about your research work. Thank you.
Okay, so thanks, Dr. Uruj, for asking about my research. First, I should congratulate you all, guys. So this is a huge participation. And first of all, I should thank Dr. Ali. Actually, he presented very well. He touched each and everything. So being a molecular biologist and neuroscientist, I know this primer designing is key in the success of each project. So I have been involved, in gene targeting, basically functional genomics work related to neuroscience. So I worked at 6 different institutes of 4 countries. So I know Dr. Ali, actually, I met him in Korea. So I know the primer designing is the key, because if I look at my work, I actually more targeted the genes, like gene knockout, knock-in, knockdown, and even overexpressing genes using the viral technologies. In all approaches, key is primer designing. So this is something very important. I think there should be a workshop, so the people should learn how to actually design a set of the primers that can make them successful. So this is key, basically. So you know that these are the innovative approaches like biotechnology. So I'm very much involved in gene targeting, and purpose is to validate function of gene. So how we do, as I mentioned, gene targeting, knock-in, knockout, knockdown, and overexpression. And once we do, then we actually check its pathogenicity using electrophysiology approach, and that could be done single cell level, like primary neurons, HEC cells, even iPSC, the induced pluripotent stem cells, and again at brain level, like the brain physiology. So we do like in mice, rats, and even in human I have done at Children's Hospital of Philadelphia. And even at bigger level, like the system level. And more importantly, I was involved in precision personalized genomic medicine. That's the future of medicine. And that's been done recently. And on top of that, I have been involved in gene manipulations, how we can manipulate the gene in utero in the fetal brain. So this is very challenging work that's been done while the baby is sitting in the uterus, and we actually approach the fetal brain and we try to either gene knockout or gene overexpression to see whether these different gene variants that we found in the human, they cause the sickness, whether they can cause the sickness at particular developmental time frame. So anyhow, this is too much regarding my work. So if you are interested, I can show you in detail at some point. I can show you how we can bridge this molecular biology work to actually the physiology and the precision medicine and gene manipulations. So I joined this forum as a member advisory board so that I could have a better picture of my region and could connect other scientists, especially those young scientists, so who are working on molecular genetics, or those who are interested in personalized precision or genomic medicine. So feel free to contact me through any source. Like I know that Dr. Ali in person, I met him, and Shahzad, who is the admin of this forum, he's very close to me. So anyhow, if you allow, I can share my information, or you can
Yes, sir. Why not, sir? We supposed to have another informative webinar from your side too. People want CRISPR-Cas9 and some other techniques to know. So we believe that you will give a nice seminar or series, be a part of a webinar series and give your information or technique knowledge to
Definitely,
us.
yeah. So definitely, I would love to share my work
Okay.
and fully done and
So,
published. Mhm.
so we can assume that you will be the next guest speaker of our seminar.
So
Webinar
yeah, so
series.
please share. Yeah, so whatever the possible slots you have, then I will check my calendar and
Okay.
I will inform whatever is working for me
Okay.
because sometime I'm busy during weekends giving talks
Yeah, no, no. Yeah,
either
sure, sir.
in Pakistan even.
Because we want to listen you. People want
That,
to listen. I
so
think.
I would love to share my work.
Okay, Rao Zubair Khalik have a question. Rao Zubair Khalik, what is your question? You can unmute yourself and ask the question. Rao Zubair Khalik. He mentioned me that he want to ask something.
To me, you want to ask question to me?
I don't know whom he want to ask, but he want to ask. He just mentioned my name and want to ask.
Okay. Okay.
Okay, and people learn more from you, sir. We got the response in the chat box. It is very informative. Please share your work. It will be very grateful. Ankita Patnaik. Yes, he will, inshallah. He will. Okay, we will definitely arrange a webinar with Dr. Tariq Zaman also. So don't worry. We will let you know in our from our webinars platform. So don't worry, anyone.
Mhm.
Okay, we
So just
are
yeah, so just a suggestion that
yes.
I will not be available until mid September. So after that, I
Okay,
can make
okay, okay. So after September, we will have a webinar with Dr. Tariq Zaman.
mid
So
Sep after mid September anytime. Yeah.
okay, mid. Okay, so we will you will let us know and we will. Okay,
Thank you.
so thank you so much, Dr. Tariq Zaman and Dr. Muhammad Ali. We have a very informative session with you both. Thank you for joining us. Thank you, attendees and participants who make this webinar session very successful. Thank you so much. Please don't forget to fill the feedback form. Okay, I have shared a lot of time the feedback form link, but still attendees want to ask about the link of the feedback form. So get the feedback form link from the chat box and fill the feedback form as soon as possible. Thank you for joining us. Signing off from the session. Allah Hafiz.